This is often the most dreaded part of this chapter. How phloem works, how translocation and sucrose moves in phloem tissue. It's not a big deal if you understand it in the exact order of the steps that I'm going to give in this segment. First, let's start with some introductory points. We have to understand what's going on. Tooth in translocation. Translocation in the phloem. Actually, I'm going to switch back to that thin. That's better. Translocation in the phloem, in phloem tissue, assimilates or sucrose. Movement of those things are going to require energy. We need ATP. It's an active process. It's an active process. Now, the phloem, we often call it the phloem sap. These assimilates, sucrose, and everything and whatnot are translocated from a source to a sink. We talked about translocation. We defined translocation properly in the previous spreads. And what was the last point about translocation we talked about? Sucrose can move in both directions. In bi-directions. Nope, not bird. Let's call it like this. Move in a bi-directional manner. You can even call it a multi-directional manner. Up and down. You can think of it like up and down, left and right, can move in any form of manner. Now, this is the part that you have to really pay attention to. What color should I use? So I'll use a green. Let's use a different green from what we've used so far. Okay, that's a brand new green. All right, let's see. What we're going to discuss right now is the active loading of sucrose. Active loading of sucrose. I'm going to teach this to you in a neat and nice three-step format. In a three-step format. If you remember these three steps with this little diagram I'm going to give to you right now, transport in phloem is not going to be an issue in any form of way. Right. Referring back to the structure of phloem, you remember phloem is made out of, first we have sieve tube elements. This diagram is going to be very important to help you understand things. I think I should use a thinner pen. Yep. Nope. Yep. Okay. First, let's draw this. Actually, let's zoom in. First, I'm going to draw like this. And remember, these things were perforated. We call these perforations. End walls were perforated. Were perforated. And then we had these special cells right next to them. And then we have just plant tissue, other plant cells, like so. And what were these parts? The first part was sieve tube elements. Sieve tube elements, and then we have the companion cell. We have the companion cell, and then just, plant tissue, just parenchymal plant tissue. Slash, say, some kind of plant tissue, and these are the general structures. And we know clearly the companion cell has a large vacuole, and it does consist of a nucleus to support all the functions of the sieve tube elements like this, and other organelles as well, like mitochondria, ribosomes, and similar things. Okay, This is the first thing you should understand. Here we have sieve tube elements, here we have the companion cell. Now, just to give you a further reference, let's go back to the structure of phloem. See, this is where we talked about the structure and function of phloem tissue, and this was the exact structure. Okay, here we have our sieve tube elements. These are the sieve tube elements, and then we have the companion cell, and then just plant parenchymal cell on the right hand side. It's the same exact diagram. Okay, don't confuse. Don't think that I've given you a secondary diagram. It's the same thing. It's just the companion cell is drawn a little bit bigger, and sieve tube elements are like this, just to help you visualize the three-step process. Okay. Right, now I'm going to do something like this. I'm going to pick this blue and I'm going to draw a small circle here. Right, it's a nice little circle. And then I'm going to expand that circle like this. And then I'm going to draw a second circle like this. And I'm going to fit it onto this as perfectly as I can. Like as much as I... Okay, perfect. Now, do you understand what's going on? It's a zoomed in image. It's a zoomed in image of the companion cell membrane. Companion cell membrane. So if I drew the companion cell membrane, it would look something like this now. We have a bilayer, a phospholipid bilayer like this, spanning all the way through, like so. And then on this, we have special proteins. I'll use a different color to draw those proteins. Okay, let's call that one protein, and then I'll have a second protein like this. Let's just give it a small different shape. All right, like so. I'll complete the membrane like this, like so. Right. Let's label this purple protein first. Now this is just a simple hydrogen ion channel. I'll call it an H plus channel. H plus channel, hydrogen channel, hydrogen ion channel. And I don't even know what this color is called, but this thing is a co-transporter molecule. This is a co-transporter molecule or co-transporter protein. Why do we need a co-transporter and how does the H plus channel come into play? Let's discuss that right now. Now, I told you that I will be teaching this to you in a three-step process, and if you understand the three steps, that's all you have to know about the active loading of sucrose. Okay, right. Let's visualize this. Now, this side is the companion cell, meaning this side is the cytoplasm of the companion cell. This side, what do we have? It's the plant parenchymal cell. That's clear, right? So this side is just simply this area. Okay. The first thing that's going to occur, I'm going to use yellow, a very bright yellow to represent this. Hold on. Yep, that's good. Right. This is the first step that happens. The cell membrane of the companion cell pumps H plus ions into the plant tissue through the H plus pump. H plus channel, or you can call this the H plus pump, like this. We have pumped H plus ions into the plant parenchymal tissue. So now this plant parenchymal tissue is going to be full of H plus ions. It's going to be full of H plus ions. H plus ions. That's step one. So let's word that out nicely. I just picked white color before it's gone. Let's pick that. White, white, white. Okay. The cell membrane. Do you want to type that? Let's type that out. I mean, no. Let's write that out. The cell membrane of the companion cell pumps H plus ions out of the cell, out of the companion cell, into plant parenchymal tissue. I'll call it just plant tissue. Okay, that's the first step. It's very easy to understand, right? H plus ions are pumped. Now, in biology, when it comes to movement, most of the time, these are the ions that are involved. H plus, K plus, Cl minus, Na plus. You'll see a lot of H plus being involved. H plus involvement means H plus pump involvement, proton pumps, and so out of the cell into plant tissue by active transport. By active transport. Why are we doing this though? Why? So first thing, we need energy. That's why this is active transport. Now the reason why we're doing this is to create a diffusion gradient, for H plus. So we need to create a diffusion gradient for H plus to come back. Why should it come back? Let's see. That brings us to step 2. Now, okay, H plus ions are outside here. I'll just pick one. Now this is what happens. This guy is going to come back, but he can't come back through the H plus channel. It can't come back through the H plus pump. The only way that this guy can go back into the companion cell, so these guys can go back into the companion cell, is through this co-transporter protein. Is through this co-transporter protein like this. Okay. But why is this known as a co-transporter? Co means 2. Okay, co means 2. So this protein is able to move 2 substances at once. The first thing now we know it's H plus. What's the other thing? In plant parenchymal tissue, what's the other thing we have? We have tons of sucrose. Plant cells, we have the glucose that was converted to the sucrose. Now we need to get the sucrose into the sieve tube element. That's why we're trying so hard. We need to take the sucrose into the sieve tube elements. It just cannot go through like this. It has a specific process, and that's the process we are discussing so far. So what happens? The H plus ions along with sucrose. The H plus ions along with sucrose is transported back into the cell. Transported back into the cell. Very simple. Back into the companion cell. So let's write it down like this. In this direction, along with sucrose, now we have H plus coming back. H plus coming back. That's step 2. Now, where's the sucrose? Where's the sucrose now? Now the sucrose was here before. Now it's in the companion cell. Is this where we want it to be? Is this where we want the sucrose to be? Not really. We want to put it into the sieve tube elements. So that would be, so that's the only way that it can move now. If it comes here only, it can go up or down or get translocated in any direction that it requires. For that to happen, we need step 3. STEP3 ... So now the high concentration of sucrose. That's how you write concentration. You should know that by now. The high concentration of sucrose now in the companion cell causes sucrose to just diffuse down the diffusion gradient into sieve tube elements. So there's a ton of sucrose that is building up here. So sucrose. Just draw a couple things. See, a lot of sucrose molecules that are building up in the companion cell because of this process. So then they just simply diffuse down the concentration gradient into the sieve tube elements. Now we have it where we want it. So this sucrose can now go up or down, left or right, anywhere through the plant and be translocated to any point of interest, and that's the overall process of how sucrose is actively loaded. This is the active process of sucrose loading. And afterwards, there's one other part. We usually refer to the movement in phloem as active and mass flow. This is an active process, active loading and mass flow. Now let's talk about this mass flow thing. What is this mass flow referring to? I'll type that out. Now, as sucrose is loaded into a sieve tube element, its water potential drops. Meaning the water potential of the sieve tube element drops. Why? Because a solute is entering. Water potential is drops. Water potential drops. Thereby, more water begins to flow in, creating a hydrostatic pressure difference which drives the phloem cell. Now, sucrose, just because it's inside the sieve tube element, is not going to move. Okay, it's not going to move. We need water. Now here we have a lot of sucrose. Because of that, there's a water potential gradient, and what's going to happen? Water is going to come by the process of osmosis into this like so. Water is going to come by the process of osmosis into this cell like this. And therefore, we have a hydrostatic pressure difference because of that. Hydrostatic means water, okay? Hydrostatic pressure means water pressure. Because of that hydrostatic pressure difference, mass flow in phloem is going to be possible. What is going to mass flow? The sucrose. The true sucrose will be translocated using the active loading mechanisms and mass flow. The closing statement for this particular spread that I will give in an asterisk form would be xylem and phloem coexist. The water is actually coming from xylem vessels. That's why we get a hydrostatic pressure difference. Where do you have xylem vessels? Xylem vessels are right next to the phloem vessels. That's why. Okay? Yep. Enough about mass flow. That is how all of this works.